In today's lesson, we're going to think about how features are inherited, and we know from last week that the genes are carried on the chromosomes that you inherit from your parents, which you would find in the nucleus of the gametes that go on to make a new individual. Now, each new individual is different, but I want you to think about twins. Okay, so first of all, do you know any twins? Are they identical twins? What differences do they have? And what features have they inherited from their parents? Okay, next thing, because we'll come back to that later about the twins, is to think about what you learnt in previous lessons. So to write down, please, in the no column, things that you learned in the previous lesson, and in the wonder column, things that you might have questions about. These would be quite good if you're coming to an in-school session. You could present them to the teacher and they might be able to answer those questions for you, I hope. Okay, so we're in the genetic inheritance part of this unit today, and we need to be able to identify characteristics that are determined by one gene and identify the genotypes and phenotypes of individuals for particular genes. Explain the key terms to do with inheritance and explain how those characteristics are determined, and also conduct a genetic cross diagram to make predictions. We usually call those Punnett squares. There are some keywords across the bottom there that you are familiar with already, I hope. Gamete, chromosome, gene, allele, genotype and phenotype. Okay, so how are characteristics inherited? Everyone inherits their genetics or their genes from their parents. Many of those genes have got different forms, so different types of a gene are called an allele. Every allele is going to code for a different protein, and those alleles determine which characteristic you have. So for instance, if we're talking about the gene for eye colour, there is more than one allele that will determine the colour of your eyes, which is why your siblings, your brothers and your sisters, may not have the same eye colour as you or as your parents. Okay, last time we looked at these words, gamete, chromosome, gene and allele, and you should hopefully be able to match these up. So I'll just give you a moment to think about that. And let's review. So the gametes are the sex cells that contain the chromosomes. Remember, the sex cells are the egg and the sperm cells. Chromosomes are those molecules that we inherit from our parents, and they contain the individual genes, of which there are many thousands. A gene is the unit that determines the characteristic. So for instance, we mentioned eye colour is a characteristic, and the alleles are the variations of those genes. So maybe blue eye colour, maybe brown eye colour in this example. So now we're going to go through the rest of the keywords, and I want you to write down the list on the left hand side and then see if you can sort out the definitions on the right hand side. So you can pause the slides while you do that. Okay, let's go through those. Dominant in terms of genetics means that you are going to express the phenotype even if you only have one copy of the allele. So you inherit one copy of the allele from each of your parents, but if you inherit a dominant allele, that will be expressed. So for instance, for eye colour, if you inherit a brown allele from one of your parents, you will have brown eyes, regardless of what you inherit from the other parent. Recessive means that the phenotype will only be expressed if you have inherited two alleles, one from each of your parent of the same characteristics. So for instance, if you have blue eyes, you must have inherited a blue eye allele from each of your parents. They might not have blue eyes, but they might carry the blue allele. Homozygous means that you have inherited two identical alleles from your parents. So for instance, blue eye colour, you must have inherited a blue eye colour gene from your father and a blue eye colour gene from your mother. So you are homozygous, homo meaning the same. If you are heterozygous for a characteristic, that means that you inherited one allele from your parent, your father, and the other allele from your mother, and that they are different, hetero meaning different. So if you have brown eyes, you could be heterozygous for eye colour. You could be homozygous, you could have two brown-eyed genes, but you could be brown-blue. So the genotype tells us which alleles are present in that organism for a particular gene. So for instance, if you're blue-eyed, you will have small b inheritance because blue eye colour is a recessive allele. The phenotype is just the physical description of that characteristic. So for instance, we're talking about eye colour again, we would be saying the phenotype for a particular person would be that they have blue eyes or brown eyes or maybe green eyes, which is a little bit more complicated in terms of inheritance. Okay, so what I would like you to do is just make a paragraph using those keywords that we wrote down on the left-hand side there to try and explain this new information. You can pause the slides and do that, please. So we've explained in the grids before on the previous slides that some characteristics are controlled by different genes and some of them are just controlled by one gene. So for instance, your nose shape is controlled by different genes. That means it might be a combination of the genes you've inherited from both of your parents. Or we can look at earlobes, where if you inherit one gene for detached earlobes or attached earlobes, you are going to show that characteristic because it is dominant. So you can draw a Punnett square diagram or a genetic cross diagram to show how that type of inheritance works. These are some examples of some dominant and recessive characteristics. Sorry, the image there is a little bit wonky. On the left-hand side there we have some dominant characteristics. So wavy hair, brown eyes, we talked about before, free earlobes, freckles, ability to roll your tongue, having a projecting chin and a straight nose. You can compare yourself with your parents to see which of the characteristics you inherited from which of your parents. Recessive characteristics, to have these characteristics, it means you must have inherited the allele from both of your parents for that characteristic. So for instance, if you have joined earlobes, you'd have to have a look in the mirror for this, or maybe feel around the bottom of your ear. If you have that characteristic, then you have inherited the gene from both of your parents for joined earlobes. Blue eyes we've already talked about. If you have blue eyes, you must be carrying a blue-eyed allele from your mother and a blue-eyed allele from your father. And we've got a few other things there. Tongue rolling is quite a good one. So ability or inability to roll your tongue is inherited from your parents. And if you cannot roll your tongue, that means you have you have inherited the same gene from both of your parents, preventing you from being able to do that. We're working now towards constructing a genetic cross diagram or a Punnett square, and we're going to use an example of detached earlobes to do that. On the first line there we have a capital D. Now a capital letter means that you're talking about an allele which is dominant. So having detached earlobes is the dominant condition. Most people have detached earlobes. The small d or lowercase d is the recessive allele, and that codes for attached earlobes. So less people will have attached earlobes because you need two copies of the recessive allele in order for you to have attached earlobes. Okay, so we've got two individuals with genotypes below. We've got the mother and the father. The mother is big D, little d, so she carries the allele for detached earlobes, and the father is little d, so he does not carry the allele for detached earlobes. Now I want you to think about what the phenotype of the mother and the father will be. So that means what characteristic is expressed, what do they look like in terms of their earlobes. Have a little think about that. Okay, so the mother carries the dominant allele, so that means she will have detached earlobes. The father is little d, so he does not carry the detached earlobe allele, therefore he must have attached earlobes. Okay, now we're going to think about calculating the chance of their children having attached or detached earlobes. So we're going to cross the two genotypes to give us an answer to that question. Now we're going to have a go at drawing the Punnett square or the genetic cross diagram for this particular characteristic. So we're still talking about attached or detached earlobes, and the Punnett square is the grid that's shown on this slide. Now in that grid, the genotype of the mother has been written across the top, and the genotype of the father has been written down the side. So the two individual alleles are split and each one is put into a different box. Okay, in order to fill the blank spaces in the box, we need to think about the interaction of the two alleles. So for instance, in this box we're going to look what the mother contributes, and that's capital D, so the dominant allele, and the father contributes small d, the recessive allele. Okay, so I want you to fill in your Punnett square now before we move on to the next slide. Okay, so here we are with the Punnett square completely filled in, and as I said, we've taken the dominant allele from the mother, the recessive allele from the father, and combined them in the new individual, so the child. Okay, so this child has one copy of the dominant allele and one copy of the recessive allele for having attached detached earlobes. And we've filled in the other ones as well. Hopefully you got the same answers as I have. Okay, next we're going to think about the phenotypes of these genetic variations. So the phenotype, remember, is what the person actually looks like when the alleles are combined. So I want you to think about what these phenotypes are for big D, little d, and for little d, little d. Okay. And then I want you to calculate the chance of the offspring having attached and detached earlobes. Now, Punnett squares have got 4 boxes here for representing potential individuals. Okay, so you can imagine that this couple might have had 4 children, and every time they had a child, there was a chance of them inheriting a dominant allele or a recessive allele. Okay, and that would determine whether or not they display the particular characteristic. Okay, so if we've got 4 boxes, and in 2 of those boxes the dominant allele is shown, that means that 2 out of 4 of these offspring are likely to have detached earlobes. Okay, so the chance of having detached earlobes is 50%. And that means the chance of having attached earlobes is also 50%. Percentages are always going to have to add up to 100%. Okay, sometimes you might be asked to give these as a ratio, so 2 to 4, which is the same as 1 to 2 for detached and attached earlobes. And that's quite a simple example. In the exam, you're normally given marks for filling out the parental alleles correctly, but it doesn't matter which way around they are, mother or father, up the side or across the top. You get further marks for filling in these boxes, and then there are normally marks available for calculating the percentages of a particular individual having that trait. So for instance, having attached or detached earlobes. This is an example for you to have a go at, to do with mouse fur. And we have 2 phenotypes for mouse fur, so this is what the mouse looks like. The mouse might have brown fur, or the mouse might have black fur. Now, if the mouse has brown fur, it must have inherited 2 copies of the recessive allele. Okay, so its genotype will be small b, small b. If the mouse has black fur, it needs to have inherited at least one copy of the dominant allele, so that's capital B. So we've got two genotypes given there. We've got big B, little b, or little b, big B, makes no difference which way around you have it, or big B, big B. Both of those genotypes will result in a mouse with black fur. Okay, so if we've got mouse 1 and mouse 2, mouse 1 is small b, mouse 2 is big B, little b, I want you to make a Punnett square and calculate the percentage likelihood that the offspring of the mouse will have brown fur and black fur. Okay, I want you to pause the slides now, please, and have a go at that. We'll come back to this at the end and just mark it. Okay, so what I want you to do now, please, is to explain in your own words how earlobes, whether they're attached or detached, and also fur colour in mice would be inherited. Okay, so we've got some success criteria at the bottom there. We've got identifying the characteristics determined by one gene, and mentioned there the genotypes and the phenotypes. We have explaining the key terms to do with inheritance and explain how the characteristics are determined. And then finally, I want you to have a go at constructing a genetic cross diagram to make predictions and talking about how that works. Okay, if you want to have a little bit further stretch and challenge, you can imagine a world in which there is no inheritance. What would we all be like? Okay, so I will set for my group a past exam question about inheritance. I imagine the other teachers will do the same. Okay, and that will be the thing that I would like submitted. Okay, and then on the little grid that we did at the beginning, I just would like you to add something new that you've learnt into that last column, please. I did say we'd come back to the twins question from the beginning of this presentation, and I'm sure that you all know that there can be identical and non-identical twins, and the identical twins do have exactly the same DNA as each other. They are essentially clones, and the reason for that is that they have inherited all of the same alleles from their parents, because the fertilized egg cell has divided in two, so there can be no other alleles in one twin as in the other. You can't have identical twins that are male and female, of course. There is one thing that is different in identical twins, and that's their fingerprints. I wonder if you know why, and perhaps you can go and find out about that, or maybe ask your teacher if you haven't managed to find out in your in-school session. And before I sign off, let's have a look at the answer to slide 28. This is the Punnett square you should have constructed. So we have a mouse that has little b alleles, and we have a mouse that has big B, little b alleles. And I've constructed the Punnett square there. It's pretty similar to the one we had for earlobes, actually. And the chance of the mouse having brown fur is 50%, which also means the chance of the mouse having black fur will be 50%. Okay, so mark that or do a bit of green pen work if you need to. Thank you very much.